Ok, vamos a ir empezando. Buenas tardes a todos, bienvenidos a esta que es la última plática de estas jornadas que conmemoran el aniversario número 30 del Instituto de Neurobiología. And now I'm going to switch to English, just to our speaker to understand. Yes. So it's my pleasure and an honor to present today to Dr. Tracy Bale. Tracy Bale, it's somebody that has many achievements in her life, her professional life, and I'm going to try to do a summary of those achievements, so to take just very short time and let her to speak to you about her research. So Tracy currently is a director for sex differences research in the Department of Psychiatry in the School of Medicine at the University of Colorado. And also she is the chair in Women's Integrated Mental Health and Physical Health Research at the Ludeman Center. And also she is professor and director for intergenerational stress and health. So her studies were done at the University of Washington, where she studied the PhD in pharmacology, and her postdoctoral work at Salk Institute at the laboratory of Dr. Wiley Bale. If you don't know who was that, was the one that discovered CERCH, that's the peptide that controls stress responses. And then Dr. Bale moved to University of Pennsylvania for 15 years. She worked there, and then moved to University of Maryland in Baltimore, where she was recruited to develop the Center for Epigenetic Research in Child Health and Brain Development. So she's been interested in the stress field since she was very young, and her research focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases. Bale brings neuroscience Weizmann Memorial Award from the Endocrine Society, the Medtronic Award from the Society for Women's Health Research, and the Daniel Efron Award from the American College of Neuropharmacology. She currently is a president of the International Brain Research Organization, and also she has been awarded as the top 10 women in Maryland in 2020. So this is only a sample of the many achievements that Dr. Bale has in her professional life, and it's a pleasure and to have her here celebrating this 30th anniversary. So thank you, Tracy, and welcome.
Thank you. So that was an overly detailed and very kind introduction, so thank you, Dr. Morales. I also want to extend a huge thank you to Dr. Morales for, and as you all know, as the Institute Director, just the incredible work that she does, largely behind the scenes and seemingly without a flaw or stress, and she does it so amazingly. She is truly an inspiration. And I hope to come back to Dr. Morales at the end when we talk a little bit about EBRO, hopefully. I also want to draw attention to the incredible undergraduates that are coming into the room and that they have walked over here to attend this seminar. Thank you all. I met with them earlier for what was one of my favorite things and truly inspiring to meet with this generation who are full of energy, creativity, and the future, I am confident, of neuroscience is incredible. So thank you all for walking over here. As Dr. Morales pointed out, I am a stress researcher, and I first met Dr. Morales when we were postdocs together at the Salk Institute in La Jolla, California, which was an incredibly beautiful location to do a postdoc. if you're looking for locales. And it was in that lab that I've always been interested in the brain. I've never wavered from that interest. And I'm largely a neuroendocrinologist, and I started my graduate training really kind of on the other side, on the more the gonadal hormone influences on the brain side. And when I started my postdoc in Wiley's lab, he's known as the endocrinologist, the king of stress. He passed away some years ago, unfortunately. He was quite an individual and an incredible scientist and mentor. But it was through that, starting my postdoc, that I really began thinking about the obvious intersection of stress and mental illness. And I think that's obvious to us all, having faced the incredible stress of the pandemic. And so I'm going to start there by kind of getting us all on the same page about stress, because we use stress as kind of a big word. We throw it around a lot. I'm stressed, you're stressed. Stress, as it relates to really lasting effects and impactful influences on the brain across the lifespan, really needs to reach a level of being pervasive, compounding, and chronic. And I met with one of the incredible undergrads earlier today who asked me a question about trauma and adversity. And I think when we start thinking about that level of stress, that the traumatic events, traumatic chronicity of events, I think many of us think about the studies on adverse childhood experiences, or ACEs, and that's something I think we've heard a little bit about from talks on Wednesday and Thursday from Javier and Scott. If you were here, that it has to reach a certain level. We're not talking about acute stresses. We're really talking about the chronic impactful, like the pandemic, as being impactful, and depending on where you are in your lifespan, what lasting effects it can have. So I'm going to hit a couple of highlights of what the kind of stress I'm talking about. As an American, and maybe as a Mexican, I don't know, this is pretty stressful to us. Right now, as the government is about to shut down again, is incredibly stressful. I will speak for Scott, who has a grant that's at study section. If government shuts down, that grant doesn't get reviewed. These are very career-based stresses, as well as the divisiveness. And so we kind of laugh when we think about this individual here, or I'm not even going to say his name, but it has really pushed many families and communities to an incredible level of stressful divisiveness. There's family members who don't speak because of what's going on. So it is really in our everyday life to be very stressed by what's going on in our politics. Social media, so this is something we talked a little bit about this morning with undergrads, and I really do want to make a statement here, because I think many individuals my age and older look at social media, kind of roll our eyes. We don't have time for it. I don't care what you had for dinner last night. You know, that kind of social media. But I make really the statement here today that how we connect globally, and I'm sort of speaking with my Ebro hat, how we connect globally, how we think about collaboration, how we think about resources, how we think about advocacy. If you have trainees you want to advocate for, if you have programs you want to advocate for, if you need to be advocated for, social media really is one of the ways that we can do that easily and accessibly. Now, all platforms aren't available across the world, and that is an issue and something at Ebro we think about. But I am a huge advocate on Twitter. And so if you follow us on Twitter, or you need to be followed on Twitter, you let me know, tweet at me. It is something that, as science, is hard, and getting your face and your name and your research to gain attention, you know, social media is one of those ways that we can best do that for each other. So I'm a huge advocate. But the stress of what social media can also do, especially for a generation, I'm pointing to some of the undergrads here, that grew up with the stress of social media, knowing what everybody's doing and everybody knowing what you're doing, and mistakes that you might make. We talked a little bit about TikTok dances and how that might follow you throughout your career. That's very stressful as well. And it has led to a lot of studies as to how social media can really be pushing this younger generation towards mental health issues around always having to be on, always having to be aware. So social media can go, I think, both ways. Climate change. I've heard several people comment on this beautiful weather and how it's a little bit different down here. We live in Colorado. We had one of the rainiest, and prior to that, snowiest winters on record. The climate is changing. It's no longer even a consideration of whether or not. It is a fact. Whether or not you fall in this camp of the individual I won't mention again, it is a fact. It is affecting us. It is stressful. How will many of our communities deal with the rising water levels, the changing climates in terms of hurricanes, et cetera? It is also very stressful, something we have to consider. And I think, again, impacting a lot of our younger generations and thinking about the world today. And then we have the pandemic. The pandemic, which is not over, and I see many individuals wearing masks again, or maybe still. I will say we just came from the Ebro World Congress in Granada, and I was very concerned that that was going to be a super spreader event because more people in my circle of family, friends, and colleagues were COVID positive than I ever remember in the 3 prior years. So it's back. There are new vaccines coming out. I'm going to advocate for, I don't care where you fall on the age lifespan spectrum. If you have access, please do get the new vaccine. It is incredibly stressful. And that period where we were locked down was so impactful. It was something we had never experienced before. We didn't know what was going to happen. There was so much stress. And again, especially I had trainees who largely, many of them living alone, were not allowed on campus, and so they were locked down at home by themselves. They didn't really have the tools and resources to know how to cope. Many of them were pushed into a very scary place with mental health. I don't think it's something that we're going to rebound quickly from. I think it's something that's going to have lasting effects. Towards advocacy, I want to remind many of my senior colleagues in the room that when you're reviewing individuals who started labs during the pandemic, I had several trainees that were assistant professors. It was incredibly stressful to try and get any resources or hire anybody, to try and get equipment. All of it was incredibly stressful, and the clock has been ticking away. And I think it's something, again, for years to come, we're going to have to think about. The social isolation that happened, I think, is particularly going to be impactful for how we think about mental health coming up in years to come. This is data from the Center for Disease Control in the US, looking at the presentation of anxiety, depression, or anxiety and depression across these bar graphs here. The dotted line for each one is where we were before the pandemic. And so we were somewhere around, I don't know, 6 or 8, 10%, right? And that's taking into account all adults. That's not children, this is just adults. And if you look at where we are now, and this is only up to December of 2021, I've seen recent data as of February of 2023, and it doesn't look any different. So the thought that we would all come out of this pandemic and suddenly our mental health would be great again is just not happening. There's a lot we need to discover. There's a lot about how these chronic, pervasive stress, our family members' health and people dying, what was going to happen to our careers, all of it, our family members, is going to have an impact. And I think that's, oh, yeah, sorry, pointer. So across the graph here, that is something very impactful we need to think about. And I think it's something that a lot of research is beginning to address, both at the clinical level and thinking about outcomes. If mom was pregnant during the pandemic, lots of studies coming out now about outcomes on brain development, but also just about if you were an adolescent or if you were an early child. Imagine if you were a baby and everybody you saw had a mask on, right? These are studies that are ongoing and I think going to be very important. But at a level of thinking about stress, wow, how does stress affect our brains in a lasting way and how we interact as a community? And imagine if you had to pile on top of that, if you come from a community where you've faced lifetime racial discrimination, other kinds of trauma, that builds as a background on how we then think about these stresses that are happening in our lives. These are very, very important for mental health. And as I said earlier to the undergrads, and again, wearing my Ebro president hat, We are facing, like no other, a global mental health crisis. And access to resources is a big concern around the globe. And as I said to them earlier, across many countries in the world, especially across the continent of Africa and other low-income, low-resource countries, that there are such limited availability for mental health. Some countries in Africa have less than 5 psychiatrists across the entire country. This is something that we need to think about, because it's not just the individual who is impacted, that it can absolutely be across generations, and I'm gonna show you some data on that. Okay, so how do we think about the brain? This is really crazy. It's hard to think about. We know in terms of drug discovery, if you were at Dr. Thompson's talk on Wednesday, you know, we think a lot about what's the next drug? How do we target this? Some of the drugs that we have are okay, but the next generation might get at this in a rapid acting way. You heard a lot about the studies on psychedelics from Dr. Thompson. How are we thinking about the brain? How did your brain, as you sit here today and thinking about everything up until the point of today, how did it affect how you interact, how you cope, the tools that you have, which all of us don't have, right? Is that about how you were raised? Is it about your DNA? Well, actually, it's really about all of the above. And Tom Insel, who used to be the director of the National Institute of Mental Health, used this equation, and I think it's incredibly important for all the research that we do, is that it's really about the G by E by D. So it's about the genes that you inherit from mom and dad. And there is not a thing you can do about those genes. Those genes determine a lot about the baseline of your risk and your resilience. If you inherit genes at risk for hypertension, at risk for diabetes, at risk for alcoholism, or at risk for mental illness, those are the genes that you've inherited. And if you're aware of them, you can sort of dictate your exercise routine or your diet or avoiding the risk factors that you know might attribute to presentation of disease, right? Mental illness is very difficult because we really can't control the pandemic or social media impacts or our environment around us. And that's the E. The E is the environment. And the environment doesn't have to be negative. It can be very positive. You can have a very impactful influence of a very strong community network, very strong social support, a very strong family support. And that can be really influential to, depending on what your risk genes are. It could also be negative, of course. The stress that we're talking about is one of those E's. But the D can matter a lot, and that's the developmental windows. So was your mom, did she experience trauma or adversity during pregnancy? Or even before pregnancy, mom or dad? Did you experience early childhood adversity? Did you experience a particular trauma during adolescence? I'm going to tell you some of these stories and how we're learning about this today. And it goes across the lifespan. It doesn't have to be just developmental. It's about the windows of vulnerability. So all of those things combined are really important for how your brain, how you interact with the world today. Underneath is SABV. SABV is sex as a biological variable. And what that means is that sex as a biological variable interacts with all 3 of these components. The genes on the X and Y chromosome, so if the cells in your body, doesn't matter if it's a neuron or cells of the reproductive tract, if they're XX or if they're XY, they respond to the internal environment differently. Because there's lots of really cool, interesting genes on the X and Y chromosome, and they function differently, and they can determine a lot about how a cell responds to the environment, all the way down to the level of how the mitochondria in your cells respond. So SABV, very important. Of course, if you're XX or XY, I'm going to be inclusive here of all genders, of how we think about societal interpretation of how we think about ourselves. If you're XX or XY biologically determines a lot about how you interact with your environment. And that can be hormonal, but it doesn't have to be. It could be a lot to do with developmental timing, that's the D. How you develop in utero, when you go through adolescence, et cetera, can determine a lot about your XX and XY cells. So that's really important to think about. And I think in the last decade, we've thought a lot more about sex as a biological variable. Okay, so all of these things are really important. They make it very complicated, though. And how do we, if we're sitting around, think of any holiday when you're sitting around with your family members or friends or colleagues, and you start talking about this global crisis in mental health and why. What is the effect of stress? What is the trauma? What is it? How do we talk about it? If you're talking about somebody at the table who's faced cancer, we have language for that. Right? We have language for tests, and we have language for treatments, and we have language for tumors and the types of cancers, and we even know a lot of the genes and why it matters. Same is true for hypertension. We understand a lot. There's some really good treatments and medications. But when we think about mental health, it's complicated. How do we think about schizophrenia or autism if we tried to explain it to somebody that had no background in neuroscience? It's hard to explain because we really don't understand. We have a lot of genes, which we call risk alleles. And that means through all kinds of studies going back decades now, we've really tried to identify what makes you at risk for schizophrenia or autism. And in fact, we know the most from schizophrenia research. The largest data sets we have are around schizophrenia risk. We have not identified any genes that are deterministic to say, like, this child's going to have schizophrenia. We don't have any. We have risk alleles. And if you add them all together, depending on the studies you believe and how the value of the GWAS data, you can get about 17% of schizophrenia risk accounted for. That's not a lot. And what is the other 83%? It's the environment. It's the environment. Environment influences your risk. And that is really where the cutting edge research is now. How the environment, and in the case of the research that I do, and in collaboration with Dr. Thompson's lab, that we're really interested in stress. Because stress is something that we can understand, and as Dr. Morales pointed out, even from back when I was a postdoc, and understanding CRF, corticotropin-releasing factor or hormone, in the hypothalamus, that pathway in your brain as a human being is identical across all mammals. Well, that makes it really easy to translate for doing preclinical studies, which is not the case across a lot of other things we might think related to these diseases. So I'm going to talk a little bit about that. So if we break it down and we start with what we know stress can predict risk. We know that stress can precipitate disease symptoms. We know that stress can exacerbate things like if you have previously had an addiction, alcoholism, smoking, et cetera, and you stopped, you're in remission or not taking your drug anymore, what is the first thing that's going to send you back to that drug? Is stress, right? You're going through a stress, you're going through a divorce, you lost your job, people will reinstate. And so thinking about what are, at a very molecular level, how can we translate either from cells grown in culture, from preclinical animal studies, all the way to the human. And so I'm going to tell you a couple little short stories today of how we think about this, going from cells and animals to what we learn, applying it to human studies, and then going back again to understand the mechanisms. Because if we can understand some fundamental mechanisms, we might come up with better therapeutic interventions, and that's our hope of how we get there. Okay, so one of the other things that we really lack in mental health is biomarkers. There really is no way today that you could go into your physician, doesn't matter if it's a psychiatrist or a general practitioner, and say, I'm feeling a little down, I think I might have depression. Your physician cannot take a blood test and say, yep, comes back positive, you have depression. We don't have any of those biomarkers. And that's a real limitation for mental health because it limits sometimes the direction you might want to go if it's depression versus if it's autism, right? So a lot of individuals with autism are on a spectrum of which it's been not diagnosed until they're young adults, perhaps. And it might be confused with something like depression or anxiety or ADHD. If we had biomarkers that could lend a direction toward treatment, if you have autism and they put you on an antidepressant, it won't work at all, right? Because they've diagnosed you wrong, and the antidepressants don't work in autism, at least in most cases. So one of the things that we're very interested in utilizing animal models and cell culture models and going into the human is, can we identify interesting biomarkers? And when I say biomarkers, biomarkers can be something that is just an indication, so functionally irrelevant, but an indication of something that's happening. Or all the way at the other end, you may get really lucky, and a biomarker could be functionally relevant. In that case, it would give you an idea for where to therapeutically intervene. Okay, so one of the ones that we've been working on for the past decade is called an extracellular vesicle. And I'm going to spend a few minutes to explain because probably not everybody knows what they are. So extracellular vesicles are nanoparticles. So they're sort of like a cell, but they're not a cell. They're teeny tiny. They are secreted from every tissue in your body in all mammals at all times. They really serve a function that's sort of like an endocrine function, right? We know that endocrine, such as estrogen or testosterone or cortisol, right, that they secrete from tissues and they act on distant sites, largely to promote things like transcription and have that kind of a function, right? But transcription takes a lot of time, and it doesn't have a great specificity because it can act at so many tissues. Extracellular vesicles, or nanoparticles, are secreted from specific tissues to have specific functions either locally or in distant sites. They're really a messenger. And the way that they do this, and here's an example on the left of the screen. Let me use my pointer. On the left of the screen here, they have a ton of proteins in this lipid membrane. These proteins are very specific from the tissue they're coming from and the tissue they're going to. So they can be secreted locally. Think of a placenta in pregnancy. There's a ton of work that's been done on the placenta. If something is happening and some cell types, right, there's lots of cell types in the placenta, and those cell types are responding. Mom is pre-diabetic, or mom is really stressed. The placenta, wanting to make sure that the fetal compartment is getting all the resources it needs, might change what it's doing. This might be a way that these cells can say to all the other local cells, let's change metabolically what we're doing so we can make sure that the fetal compartment is going to get what it needs because mom is in a different metabolic state. That's just one example. Okay, so they can act locally. The placenta also secretes a ton of these nanoparticles in pregnancy into circulation to make sure they act like a homeostatic regulator. Think of your house thermostat, right? Suddenly it's cold out or suddenly it's very hot out. You're going to adjust that thermostat. And that thermostat sends signals throughout the house to turn up your air conditioning or turn it down, et cetera, turn on your heat. That's kind of what these are doing in circulation. They come in all different flavors, and that's really important for their function. So a given cell can secrete all different kinds of nanoparticles. You've probably heard of exosomes. That's one type of EV. These are really tiny and very small, and they come from a very specific intracellular compartment. There's also microvesicles. These are much bigger, are likely to contain parts of the actual cellular membrane when they're secreted, et cetera. There's all different kinds. We're discovering new ones every day. And so they're secreted, like I said, including from and within the brain. There's a lot of research. So typically what happens in science is that cancer biologists, who have done the most work in this field, make these huge discoveries. In fact, they've figured out that these biomarkers can be used for detection of all kinds of cancers, right? So they've decided this is a way that we're detecting the recurrence of cancer or whether the therapeutic intervention is working. You can make synthetic nanoparticles now, and you can put in them drugs you want delivered to a specific tissue, right? It's a way to give specificity to your treatment. So cancer biologists have done a ton of work. But when it really gets sexy and when it really gets exciting is when neuroscience gets involved. And that's really where we are now with extracellular vesicles. Neuroscientists have figured out that these things communicate within the brain and between the brain and the body as well. And so that's something that the composition inside of this nanoparticle is all kinds of things, parts of mitochondria, there's all kinds of RNAs and DNAs. Largely what the function is, the major function, are small non-coding RNAs, like microRNAs, for example. And so when these particles, nanoparticles, get taken up at a distant site, the placenta secretes it into circulation, mom's pre-diabetic and her glucose levels aren't being regulated well, it will increase the specific nanoparticles into circulation, and they can act either at the liver to turn down gluconeogenesis, they can act at skeletal muscle to say we need to uptake more glucose. So the way that happens is a little bit of protein-protein interaction, which I'm not going to get into, but a lot of small non-coding RNA or microRNA that get taken up into the cells, and those microRNA can immediately target mRNAs to be degraded so that you're not translating into proteins. It's a way faster mechanism than waiting for the slow grind of transcriptional changes to happen. So that's a lot of what happened. On the right side there is just telling you some of the research that's been done in these nanoparticles. They are very fast. The turnover of nanoparticles based on signaling homeostatically is very fast. So if I were to suddenly, if there was a huge alarm that went off and everybody was startled and stressed, you would see pretty rapidly a change in the composition of the nanoparticles being secreted into your bloodstream. They can be local or systemic, and the fact that they're so dynamic makes them a really interesting potential biomarker. Because we can ask questions systemically of you as a human being in a way that we can't do with other markers related to mental health. We can ask questions of, if you have had adversity early in life, you may interact with stresses in a way that everything becomes stressful in your environment. And we can look at those changes, not just at stress hormones, but the other types of signals that could tell us specific tissues that may have been reprogrammed. And that's new knowledge that we didn't have before. Okay, what do we measure when we measure EVs? We can do them in mice, we can do them in culture, and we can do them in humans. And there's a couple different things of information we can get at a higher level, and we can get at a very low targeted level as well. So on the left is looking at vesicle size. Like I told you, they come in all flavors. We're learning more and more about how they're programmed within the cells, how changes in mitochondrial function within certain cells changes the dynamics of how they're secreted, et cetera. So like I said, we learn a lot about taking EVs from your blood. We can precipitate them in all different kinds of ways. We can identify the specific microvesicle versus exosomal populations. And we can do that by, one of the ways that we do this is by nanocyte technology, which uses Brownian motion, how things move, to determine these sizes. We can also look at their zeta potential, which is just another way of saying the overall charge composition, which is based on both the size and the types of proteins that are important. And that's really important for the following reasons. If your zeta potential changes and the overall population being secreted is more neutral in charge, it means they're going to aggregate faster. So if you want a signal that you want to be very dynamic but to turn off, you can make them more neutral. They will aggregate and they'll be taken out of circulation very quickly. If you want the signal to stay around longer, you make them more negative in charge, and they will repel each other and stay in circulation longer. So their stability and their rate is very important. On the left here is looking at the size, and that also can tell us a lot of information. When you shift, we know in stress across all species, the glucocorticoid levels associated with stress, whether it be chronic in nature or acute in nature, will shift toward more of an exosome, a smaller nanoparticle in circulation. We don't know why or what they're targeting yet, but we know that we see that across many species. And this can tell you a lot, again, about what is going on, about how the nanoparticles might be targeting and what they're taking up. So at a very big level, a very large view level, we can learn a lot about what the changes might be. We can also take those nanoparticles and we can sequence the RNAs, and we can say the composition of what is being targeted is changing. We can learn, because we know through alignment what types of genes those microRNA might be targeting. And through the proteome, doing a proteomics assessment, we can say which tissues might be involved. Okay, so that's a lot of information we can gather. And I want to highlight that we're doing this across the lifespan, because we're interested in how this might affect everything from how fertilization may come together, sperm and egg, because you're not going to get a brain if you don't first get fertilization, right? So what are those signals that you might bring together from mom and dad's life experiences, right? How did they shape the reproductive tract, extracellular vesicles that are communicating to their germ cells? And I'm going to tell you a little story about that. But this goes all the way up through pregnancy. As I told you, the placenta is an enormous secretor of extracellular vesicles. A ton of work has been done in this field. A lot of studies looking at biomarkers of things like preterm birth and preeclampsia, et cetera. So what do we learn? Well, we also know that pregnancy across the lifespan, I should mention, that the concentration of EVs goes up 4 to 5-fold in mammals. So there's definitely something going on with the secretion of these vesicles. But it's important across the reproductive cycle and as well as across the lifespan of what we might learn. So one of the studies that we've been doing is trying to understand, as a homeostatic regulator, might glucocorticoids or might the interaction of adversity in your life shift the composition of EVs? And if we can learn from our culture models and our preclinical mouse models Then can we take this into humans, and what can we learn from that? So we started off by thinking about, well, if the concentration of EVs goes up so high in pregnancy, what is that telling us? Can we learn something to apply for a different, if mom has pre-diabetic or has gestational diabetes, or mom has had adverse childhood events? There's lots of questions we can ask, because we know that pregnancy is a very dynamic period and a very stressful physiological period. And so asking questions about what these different cell types in this tissue, this very endocrine and sex-specific tissue in pregnancy, right? So because the placenta is formed out of the trophectoderm, right? So you have fertilization, then you have an embryo, 2-cell, 4-cell, 8-cell. You all remember from your developmental biology class. Eventually you end up with a blastocyst. That blastocyst has the inner cell mass, which will become the fetus, right? The outside of that blastocyst is trophectoderm cells. So it's coming from the XX or XY cells, that trophectoderm, that becomes largely the invading cells of the trophoblast of the placenta. So placentas, with the exception of the maternal tissue, the decidua, right, that's going to be XX, it's mom, but the rest of the placenta and a lot of the really important function is sex-specific, in that it is XX or XY. And those cells respond very differently to the changing maternal environment. And that's a huge, huge study of why so many neurodevelopmental disorders may be male-biased in presentation. I'm not going to get into that today, but we were really interested in trying to understand the biology here. So Hannah Zierden, who was a postdoc in the lab and now is an assistant professor at the University of Maryland, just began asking some very fundamental questions that were not known. One is, and so if you look at this first example here, this on the y-axis is the concentration of EVs in circulation in a mouse in pregnancy. She's comparing a non-pregnant animal here, so you can see the concentration is low, as I told you. In pregnancy, this is mid-gestation for a mouse, later gestation, and right before birth. And so as you can see, it goes up 4 to 5-fold in concentration, just like it does in humans. On the right, what she was asking about this concentration of EVs in circulation was, what is the effect if we stress mom? This is mice, not humans. Stressing a mouse, very easy. Mice, not being the smartest critters in the world, will react to just about anything. They're very easy to stress. And so every day, early in gestation, which is the equivalent of the first trimester, we're stressing mom, and we're asking questions across the remainder of pregnancy. And as you can clearly see, that concentration goes down dramatically and remains down. So that starts to tell us something biological at the placental level that Hannah is now continuing to examine in her lab at College Park. Asking those questions of, okay, now we know a tissue that's likely involved because it is profoundly important for secretion and communicating with mom in that compartment to really preserve fetal health. So what is changing biologically at that level? And one of the things that she examined was just a direct effect of glucocorticoids. We know that glucocorticoids are not everything about stress, but they are the main factor. They are the stress hormone that is produced in all mammals. So she began asking this question again by asking, is it changing the types of EVs or the concentration of EVs over time? And then this is over, just looking at the circadian rhythm. So in all mammals, across your rhythm, whether you're nocturnal or diurnal, you're going to have a huge change in your cortisol in humans or your corticosterone in mice. Those levels will transition to levels equated with stress throughout a 24-hour cycle. And so if they're changing naturally, we can ask this question of how this relates to EV concentration. So what I'm showing you here is the concentration of EVs across the circadian rhythm. Mice are nocturnal. So you're showing early in the morning their glucocorticoid levels, which are on the y-axis here on the right side. Glucocorticoid levels are very, very low. EV concentration is very, very high. But you can see over the 24-hour cycle, as glucocorticoid levels go up for these animals, the EV levels go way down. So now Hannah's been able to identify another biological aspect that she can pursue at the cellular level. How is this happening? How are these changes happening in the non-pregnant and pregnant animal at the level of a cell secreting EVs? Why is the concentration dependent on circadian rhythm? And we know that the circadian rhythm is so important for mental health, all the way up to the mental health level, but in all cells in the body, there is a rhythm. and that rhythm is largely established through how these glucocorticoids are secreted. So that's something that she's also following up on. And in a really primitive study that she began doing, she asked the question, if I take EVs, if I isolate them from a pregnant animal when the concentration is so high, and I inject them into the tail vein of a non-pregnant animal, because we know one of the aspects related to glucocorticoids on the opposite end of the spectrum for what glucocorticoids do is regulate glucose, right? They're very interrelated. And so she asked, if I take the very messy levels in a fasted animal of glucose levels, and I take those concentration of EVs, the effect that I can have is to really dampen the high levels that they'll go to and the variance of what they'll do. And so that tells us something acting about these EVs, possibly because they're isolated in pregnancy. That's something she's still doing. But because the concentration in pregnancy is so high, and the evidence is so strong now in maternal gestational diabetes, that there's something about these signals where they could be acting in lots of places, affecting gluconeogenesis at the level of the liver, shut that down, affecting glucose uptake at the level of skeletal muscle, increase the uptake, increase the response of insulin, et cetera. So those are studies that she's following up on. So that gives you a little flavor for the importance and the homeostatic mechanisms of what these very dynamic nanoparticles are doing. But as I told you, we're really interested across the lifespan of how we might use them as biomarkers of cells that have been programmed, meaning adversity early in life is one example. So you experienced high levels of adverse childhood events. You're going to have a new allostatic set point for many of your cells in the body. They're going to respond differently throughout your lifespan. So when you encounter acute stresses, they're going to seem bigger to you. When the pandemic happens, it's going to feel bigger to you. And so your cells don't care what type of stress it is. All they care about are the signals. One of those signals is glucocorticoids, right? So the cell cares about that. If it's out of sync with the rhythm, because remember I just told you that your circadian rhythm, just like a mouse's, that it goes up and down in a 24-hour cycle. So how would a cell know that it's stress? Because it has to be high levels, persistently out of rhythm with your circadian rhythm. That's when a cell knows it's stress. And it begins to shift responses of the cell. That can be true for a neuron. That can be true for, again, a reproductive cell. That can happen acutely, and the cell will return to a baseline when your stress is over. So final exams, that's very stressful, right? But you go home after final exams, your mom makes your favorite foods, you sleep a lot, you recover, your cells return to a baseline. But the pandemic, that was a long, chronic stress. And what happens oftentimes to cells, especially if they're at a different baseline to begin with because of adversity or discrimination or other types of trauma, that those cells will be responsive and have a new allostatic set point. So now your thermostat can't go back down. Your thermostat is set at a new point. And we're very interested in trying to identify what keeps our cells at that point that makes them so sensitive or so responsive. And those are some studies I'm going to tell you about. So I'm going to tell you about two studies, the first of which is going to start at the human level and go back to the mouse. And the second one is going to be a little more between the mouse and the human. So, and thinking at the human level about biomarkers, through many different conversations that I've had over many years of my career about that D, the GED, that developmental window, there is known data that says that women who've experienced high levels of adversity or trauma during a period of pubertal or adolescence, depending on what studies you look at, during this dynamic window, are at the highest risk for presenting affective disturbance. So that means depression or anxiety, et cetera, across their lifespan, right? So this is the group that has been talked about the most in the mental health space. And so we were very curious, and we've been doing studies over the past 2 decades trying to identify, well, why that window and why that group? And so we did a collaboration with the Grady Trauma Project. The Grady Trauma Project was started by Carrie Ressler. Carrie Ressler is an MD PhD who is in psychiatry residency at Emory. Emory University is in Atlanta, Georgia. Atlanta, Georgia is a majority black city. It has an inner city hospital called the Grady Hospital. And Carey was very, very savvy when he was a resident. He was seeing a lot of patients. He's very interested in PTSD, or post-traumatic stress disorder. He was seeing a lot of individuals coming through across the lifespan and with a lot of baseline high levels of trauma in their life. And he made the smart decision to say, we're missing out on a ton of data that we could learn from all of these individuals and the traumas that they've been exposed to. So he started what's called the Grady Trauma Project. And that is everybody who comes into the hospital for any reason fills out a survey. And if there's any current study going on around anything about what you've been experienced or what you've exposed to or what you're presenting with, they may enroll you in a study. And a lot of these studies will have money, they'll pay individuals to participate. It may just be gathering some of your information. It may be gathering biological material like blood plasma. It may be asking you to come in and participate in a PTSD study where they do things like looking at skin conductance and how you respond to things like an air puff, et cetera. So they capture all of this data. It's an incredible resource. And so when Carey Ressler was at this institution for a long time, but he eventually was recruited to Harvard, and he runs the McLean Hospital now. Tanya Jovanovic took over that project. And then she was subsequently recruited away to the city of Detroit, which is in Michigan, another majority black city facing a lot of trauma and adversity and discrimination. And she had started the Detroit Trauma Project. And so we do a lot of collaborations. And we started at conferences, we would get together and we would have a glass of wine and we would say, you know what, how do we address this question about women experiencing adversity in adolescence? How do we address the biological underpinnings that might give us insight into new treatment modalities, biomarkers, therapies, et cetera? And so Tanya said, well, let's look at, in our database, so we didn't recruit anybody for this study. We just used the information that was already in their database. And we said, okay, what's a specific type of trauma? Because if we ask individuals with lots of overall adversity, it's very wide and vague, and recollection can be full of error. But experiencing interpersonal violence, specifically sexual trauma, tends to have a very defined time. And so we used that information in their database to pull together a large group of women who are fully adult. So these women in this study are in their late 30s and early 40s. They're almost all African-American women. They have an overall high level of adversity in their community, but we're specifically asking about the timing of when they experienced sexual trauma. So we have 4 groups. We have women who've experienced sexual trauma in childhood, so prior to adolescence, only in adolescence, only as an adult, or no sexual trauma at all. So we're looking at the timing question. It's all women, and they're almost all African American. Okay, so in most cases, this adverse experience that happened, this interpersonal violent experience, happened decades earlier than when they've come into the hospital and given this information. Okay, so we took all of the data, out of the database, and we began asking at a higher level about health. We looked at 2 particular types of health outcomes. We looked at health outcomes related to diabetes, obesity, and hypertension, and we looked at health outcomes related to mental illness, or especially PTSD. So on the left side here, what you're looking at is dividing into our group's initial assessment statistically of no sexual trauma versus all sexual trauma groups together. So you can see all of the ends in these groups are around 25 to 28 in each group, okay, in each age group. And so you can see overall, there's an increase in body weight, hip circumference, all of the indications of earlier onset related to diabetes and obesity. And that's not surprising because that has been shown again and again, especially with childhood adversity. On the bottom is the risk for entering into hypertension stages that are more severe. So hypertension stage 1 and 2, et cetera, right? So what you can see here, again, overall sexual trauma enters in earlier. And as you can see here, you can see a shifting, especially if that happened before adolescence. Again, repeating a lot of data that's already out there. So what this data told us overall, if you look at the differences across the groups, childhood adversity, so earlier than adolescence, was really the push for entering in largely faster aging. So we didn't do telomere length, but the data strongly supports that childhood adversity makes you age faster. And so these women seem to have been entering into this higher risk group for health related to diabetes, obesity, and hypertension. But when we started looking at the data related to mental health, especially PTSD, on the right you can see something different. And that again, sexual trauma overall elevated the risk for PTSD, and changes in, this is looking at fear potentiated startle and skin conductance. You can see a shift there that seems to be driven largely by sexual trauma happening in adolescence, more specifically. Again, this is all published. This is just a snippet of some of the data that we have in examining the health outcomes of these individuals. But what I wanted to emphasize here is that D, again, the developmental timing of when a particular insult happened. And so I want to transition then through a particular aspect of the data that we found. I'm not going to go into all the other data in this paper, but we wanted to ask about the EVs. That was really where we wanted to go in the study, about trying to isolate biomarkers, novel biomarkers, that may give us some insight based on the cargo in those EVs that was either a new tissue type or a new pathway, et cetera. Okay, so what we did is we isolated all of the EVs, so the blood samples which had been banked at the Grady Trauma Project were shipped to us in Baltimore, blinded. We have no idea what the samples were. They're all de-identified, of course. We isolated the EVs from it and did all of the assessments. One of the assessments we did was an unbiased proteomics approach. And what that means is we took all of the samples and we wanted to identify by proteomics all of the proteins present across all of the extracellular vesicles in these groups. So on the top here, you can see no sexual trauma compared to overall sexual trauma. So yes, in fact, there was some big differences in the composition, the protein cargo that we detected. And so over on the right are some of the proteins I'm sorry that's so teeny tiny, but there's a couple of proteins that were just dramatically different that are shown over here in terms of those that were really low versus really high versus really low. And you can see across all sexual trauma groups, those 3 age groups, that there's a log-fold reduction in the presence in some of these proteins, annexins and galectins. Why is that interesting? Because there's a lot of proteins that were different. Because we're interested in EVs, that's very exciting, because these are proteins that are really important for determining the types of extracellular vesicles being secreted. Now remember, we're taking these from blood plasma. We have no idea the tissues that they may be coming from. And these are individuals that have come into the Grady Hospital when they were recruited back. Right? They were in the hospital for some reason. They entered into a study. They came back in to perform some of these studies around PTSD, and then a blood sample was taken. I'm going to point out that coming back in to do a study and doing the fear conditioning may itself have been a stressor. So some of the data that we're seeing is probably likely related to how those individuals are responding to a stress. So that's interesting and something that we're following up on. But I just want to quickly talk about this data. If we pull out all the proteomics by when you experience the trauma, focus on this difference right here of adolescence. These proteins that are really, really abundant, they're in blue, meaning they're really abundant, in fact, almost undetectable in the other groups in some cases. So what are those proteins? Well, those proteins were all related to keratins and keratin-associated proteins. So anybody who does proteomics knows that as soon as you get keratins in your samples, you toss them all out, because that means garbage. Somebody who was doing the pipetting got keratins in there. And so that's what we did. We tossed them out. And then I got a phone call from the mass spec facility who was looking at the data, and they said, hold on. You're only detecting this in one of your groups. You should look closer at this. So I began spending many, many late nights during the pandemic poring over this data, because it's only these proteins here in this dot plot. All of these proteins were only detected in this one group. And right, these are women that had been recruited over a limited time of a few years, having nothing to do with the sexual trauma. So it wasn't like they recruited specifically. They were just in the database. So it was only this group. So I started thinking, well, what do these proteins, what do these genes have in common? And this is where epigenetics comes in. So anybody who's interested in how genes that might show up in a particular group, particular trauma group, et cetera, you always wonder when you have a group of genes, well, how did that happen? How are they regulated the same, especially if they're on different parts of chromosomes or different chromosomes? So what came out of this that was super exciting was that the majority of these genes we identified are all part of a gene cluster called 17q21. That is super exciting because gene clusters tend to be regulated similarly. So now it tells us something about that G by E, that there's a cluster of genes that are regulated similarly that may point to something about why and when that we need to find out. Okay, so our reviewer also pointed out for us that 2 genes identified in the GWAS for PTSD, so genes that are risk alleles for PTSD presentation, are the corticotropin-releasing factor 1 and MAPTau. Both of them also in this gene cluster. So this gene cluster is really interesting and something that I knew nothing about. I'm a neuroscientist. I dabble a lot down in reproductive spaces, but certainly nothing around skin. But I was reminded back to when I was a postdoc that we began identifying new stress-related ligands and receptors, that there's a lot of the same ones, a lot of genes that are important for your brain stress response also exist in the skin and hair follicle. And there's a lot of redundancy there. So that becomes interesting as well. Why is it that there's this redundancy built in, that your HPA stress axis actually exists also around the hair follicle? Super interesting, right? And also, these women experienced interpersonal violence or sexual trauma. And we start thinking about analogies that we use in our community spaces, such as, he got under my skin, he made my skin crawl. How do we think about stress and trauma related to skin? It's the biggest barrier, right? The biggest organ we have to prevent our interactions with or detect interactions within our environment. So that's also really interesting. So we began thinking more and more about what these genes have in common, what this cluster and its relevance, and thinking about how our skin could possibly be interesting and related to things like how interpersonal violence integrates into risk for things like PTSD. And so the skin being the biggest barrier, we know the skin is highly innervated, right? It's highly innervated. It's very important for how you experience your environment. When I began digging more into those genes, especially those that are around the 17q21 cluster, something else popped out, that the differentiation of a keratinocyte in your skin into a very specialized cell called the Merkel cell has a lot of those very genes are important for that differentiation. And that was really exciting because for everybody who remembers what happened sort of during the middle of this pandemic in 2021 was that the Nobel Prize came out to Artem Patapoutian, who had just gotten the prize for identifying the piezo-2 channel, which is the transducer of light touch in these Merkel cells. These Merkel cells are incredibly fascinating if you haven't learned about them. They're mechanosensory skin cells, but they're not everywhere. They're in very specialized places, like in your fingertips. This is also true for mice, by the way. What I find fascinating is that they're in your lips. And then I started thinking about intimacy. And why is it that in intimacy we kiss? I started thinking more about what role in violation of intimacy, in violation and adversity and trauma, might that be important? It's a fascinating question. They're on other places in your skin, including your back and your fingertips, your lips, and in genital regions as well. So you can start thinking about how we may have stumbled onto this because of this particular type of trauma. So they respond to light touch. When they're activated, they're the only neuronal-like skin cell we have. When activated, they release norepinephrine that binds to the beta-2 adrenergic receptor. And that activation activates the A-beta sensory fibers that go back up to your cortex. So there's a lot to think about here. And what got me really excited as I was typing away on the grant thinking, oh, this is so cool, is that in addition to all the work, there's a lot of papers from Artem Patapoutian. You can see that they've been all over Neuron, Nature, Cell, right? These are really big fundamental papers that have laid the foundation for our sensory system, so important. Ellen Lumpkin's group also did a bunch of work, and she's a very neurodevelopmental lab. And she identified the fact that the differentiation from keratinocyte into these Merkel cells, the final stage of this differentiation occurs in adolescence. So that was where my light bulb went off of maybe that's the D for why we're seeing this in these women, that this adverse experience had happened in adolescence and established some kind of programming in these cells, which is super exciting. So now we're back to the G by E by D example I gave you. We've identified this really exciting G, this 17q21 gene cluster. We've identified a particular trauma event that happened, and we've identified adolescence as this window. So this gives us a huge amount of information and a way that we're pursuing this in a very large funded grant from NIMH that they're very excited about. And for neuroscience to then go out to the sensory system and come back again and how we might do this. That we're collaborating with Tanya again, Tanya Jovanovic, in Detroit at the Wayne State. And it's a way of taking a cohort that she has of subjects who were recruited, moms in pregnancy 33 years ago were recruited in Detroit. Their children are now 30 years old, and they're having children. So now we have this massive cohort, hundreds and hundreds of kids that were born 30 plus years ago, part of this cohort. But the beauty is that they know everything that they've experienced because they've followed them for 30 years. So they know everything that happened in pregnancy and everything that's happened up until now. And so it's a wealth of incredible information. We feel incredibly privileged to have this community investment. And it's a way in which, and I know Dr. Morales pointed out that I'm very invested in ensuring that the information we capture from this community gets back into the community that has provided the information. And so we're going to take all that, and as well in the grant, in addition to the humans, we have mice. And this is one of the ways, I'm not going to spend a ton of time on this because we're just starting on the mouse aspect, but it's a way that we can use chemogenetics, and I saw some great chemogenetics posters in the last several days. This is a way of saying, do we think these Merkel cells are part of your stress system in a way that we didn't know before? That would give us a new target, right, if they're valuable. Or are they just a part of, did we detect them because they were programmed during this window? There's a lot of questions. Mice are a great way to genetically assess that. So we can take Cre-responsive animals that have an enrichment in Merkel cells of Cre expression because of the Cre driver, KRT14, and we've bred them with DRED-GI and DRED-GQ. What that allows us to ask is in stressing the animals in adolescence, right? We can stress them. I told you mice are very easy to stress. We can give them all kinds of stresses in adolescence, and we can also give them CNO, the chemogenetic drug, to our GI mice. And that has the DRED receptors in the Merkel cells will be activated during stress. So now we're doing stress, but we're not activating the Merkel cells. Right? So we're eliminating that. So that will tell us a lot about the sufficiency and necessity using these two aspects, because the GQ is an activational DRED. And so not doing stress, just giving the animals CNO during that same window, we can ask about the sufficiency, right? So we can ask about the necessity and the sufficiency of what these Merkel cells are doing as an environmental interrogation of how stress programs, are these cells involved? We can also use a ribotagging assessment that we've put on these transgenics animals as well, that this allows us by tagging the ribosome, now we can cut out the specific tissues, we can stress them during adolescence, et cetera, and then as adults take out those tissues, and we can use ribotag to assess the active translatome to say, did it program them differently? Was it something activational that's long-standing, or is it a direct activational change, right? This is the way that we can assess this. And then, of course, we can look at sensory sensitivity. One of the ways we can do that is looking at a von Frey, like look at the sensitivity of those Merkel cells. Are there more Merkel cells? Are there more piezo2 channels? There's lots and questions to ask here. So that's how we're going to go between the mouse and the human and the mouse, and hopefully be able to assess some mechanisms. So I'm going to share with you just a major preliminary piece of data that postdoc Austin Corrigan just did on these. These are the KRT14 Cre animals crossed with the DRED-GQ. These animals are adults. They haven't been stressed. This is just asking, what will happen if you activate your Merkel cells just out of the blue? You're walking along the sidewalk, and all of a sudden your Merkel cells are activated. Is that a positive thing? Does it feel good? Or does it feel weird because it came out of the blue? Or is it a negative thing? Is it an itchy thing, a painful thing? These are all sensory aspects that we don't know yet. Okay, so what I'm going to show you when I activate the movie, these are animals that, well, actually, you know what? I'm not going to tell you which animals are which. So the animals have been fed CNO. We actually put it in a cookie treat. The animals are either DRED negative or DRED positive. Okay, so they only have, the only difference in these animals is whether they have the DRED receptor in the Merkel cell. They're all going to get the CNO drug, and we don't inject them because injections to animals is very painful. We instead give it to them in a little tiny cookie treat. And so they eat it, and then they get the drug that way. Okay, so it was given to the animals 25 minutes before at 2 mg per kg. I want to, you see what, knowing what normal mice do when you're filming them, going around in their cage, take a look at which animals you think are DRED positive. Okay, I'm going to, wait, okay, there's those. Any guesses as to which ones are the DRED positive animals? Any guesses? So these animals are behaving like a normal mouse. They're just walking around their cage, because you just moved the cages to film them, right? So what's going on? You notice that these animals, what they were doing? They're all grooming. They all went back in their nests, and they're all grooming. And that, when mice get anxious or stressed, that's the first thing that they do, right? They just start grooming, is what they do. But it's interesting, I think, that that stimulation was at least something that they noticed, that it was something, again, is it a positive experience or negative? We do not know. But at least we know with CNO activation that these animals were provoked to groom. We'll see what it means. And now we can use this assessment to look at the timing of when dread receptors are activated. Okay, so the last story I want to quickly tell you about is looking at sperm-egg interaction. And because we are really out of time, I'm going to skip through a lot of this. This is really asking the question, if you have a lot of adversity in your life, the signals that you might bring together as a male or a female sperm and egg are really important for the course of maturation. And I'm just going to give you the punchline now, and I'll just show you some of the data because we're out of time, which is that these signals coming together, largely delivered, especially for the sperm side I'm going to tell you about, are through a lot of maturational processes that the EVs signal to the sperm. This is where this field has gone for 20 years trying to assess what the signals are between generations. And really what the signal is about the environment, and that you either need to speed up development or slow development down. That's really the big picture. And if there's been a lot of adversity, the pandemic, a lot of adversity in the environment tends to slow down development and tends to reduce fertility. It's after the stress is over that you see this rebound effect, baby boomers. You see a rebound effect of fertility, right? Individuals who have a lot of stress trying to conceive, stress levels go up, fertility rates go down. A lot of individuals, when they adopt, releases that stress, and then oftentimes pregnancy will happen. And so there's a stress interaction with fertility, for instance, and that's related a lot to the environment. And so we know a lot of epidemiological evidence around the Dutch hunger winter and Swedish famines. There's a ton of evidence that these effects can be passed on across generations and affecting brain development. So this is really a summary of what the last 2 decades of research from many labs, including ours, has come down to, is that these effects don't happen during spermatogenesis. They don't happen, and that's for good reason. You would not want the environment to affect your germ cell development. Why? Because then you'd have DNA mutations and you'd wipe out a species. And in fact, they're very protected by the testis barrier here. But once the spermatogenesis ends, sperm are pushed, they cannot swim, they can't fertilize. They're actually pushed by a fluid motion into what's called the caput of the epididymis. And this is showing you a cross-section of this region. This region is critical for sperm maturation. And what happens during this time where they're put into a quiescent state, and they're receiving all kinds of growth factors and interacting with really essential extracellular vesicles, being secreted from these somatic cells, which are called epididymal epithelial cells. So it is not an environment direct action here. It is an environment action at these cells that tells the germ cells what information they should carry to fertilization. So as stress has been happening and is now mostly resolved, and a state of allostasis now has happened in these cells, I'm giving you the whole picture in one slide, that state will produce differences, as I told you before, into the environment. And one of those things it does is to program sperm differently. So I don't have time to go into all this data. I've had some amazing trainees. Jennifer Chan developed a stress in the dish model to really ask the question about what is happening at the level of these cells. So she could culture epididymal epithelial cells from mice. She could expose them to all different types of stress, glucocorticoids, do all kinds of assessments, which I'm not going to go into, to really equate that to what we can show in vivo in mice. And in that, she can collect the EVs. You can't do that in vivo because it's impossible to get into the epididymis. Okay, so she can collect the EVs and ask all kinds of questions. Nicole Moon is an MD PhD student in the lab, has really gone down very deep into the cell and asked the question about what is this new thermostat set point epigenetically. And what she's done is what's called an assessment of cut and run, which is sort of like a fancier ChIP-seq, but way more accurate. And she can show that there's huge changes in one of the major H3K, so this is histone 3 lysine 27 trimethylation. It's really a transcriptional repressive mark that has changed in these cells long after the stress is over, right? So that's what we're looking for, these changes. And to give you the punchline of where it goes all the way down into the cell, it's always about the mitochondria, because they're the energy warehouse of the cell. And if stress has changed in the environment, you might want to change your metabolic set point, and that's exactly what she found. I'm not going to go into all of that data. But the most convincing data that has come out this in terms of causality, right? The field is full of associations. The causal study that Jennifer Chan was able to do was she was able to collect these EVs in her stress in a dish model, and she could take either the vehicle, no stress, or stress EVs. She could take a male mouse and collect sperm from this mouse, so you're controlling for that environment. This is a control non-stressed mouse. Divide it into 2 pools. Incubate with the control EVs, incubate with the stress EVs, right? So that's the only difference. Wash those sperm, perform intracytoplasmic sperm injection. So these are eggs from the same female that have been superovulated, so controlling for that environment. So eggs from the same mom, sperm from the same dad. The only difference is what those sperm were incubated with. So we're mimicking that incubation that would have happened in the caput region. Then you can take the same environment to transfer these embryos into, and beautifully with these studies, you can transfer one control onto the left side and the stress EVs sperm onto the right side embryos, so you're controlling for that environment, and then you can ask mid-gestation, is there any difference in the rate of development of the brain? And you can also let them grow up and ask, how did they respond to their environment? And the take-home message was, we found huge, beautiful differences, which is that punchline that I gave you, which is really changing the rate of development. And for bringing more individuals into a community that has been devastated by war or famine, that's great, right? You want to speed up development, but at the price of what in the brain? And that's something we're currently looking at, is that the brain is much more sensitive to developmental rates, much more sensitive to pruning and establishing synapses. And you can also see that these individuals, if you look down here, as adults, they're less responsive to their environment. So acutely, they respond very differently when they're adults. So that tells us a lot about how these brains have changed, all because of the signals that those EVs were able to transmit. Okay, so the last thing, which is very cool, that this data that Nicole just developed, so I have to show you, is she's been really asking this question about, so what do the EVs do to the sperm? What is the consequence functionally? And so the 2 things you need to know about sperm, if you were to ask this question about how would you build a better sperm if you had devastation to a community, war, famine, pandemic, how would you build a better sperm to propagate your species, right? What are sperm? Sperm are 2 things. They're dad's DNA and the motors to get it to the egg. That's really what they are fundamentally. So what would you do? You would make it swim faster, right? You would make it swim better and faster, and that's what you would do to outcompete. So in fact, what Nicole did, and repeating a lot of the studies that Jennifer Chan did, and asking really fundamentally about the sperm, what this data is showing you overall, looking at respiration rates of those sperm, is that what it does is it changes the respiration rate of those sperm. And so what it should do is make those sperm more motile. And that's exactly what it does. It makes the sperm more motile. So if you have millions of sperm competing to get to the egg, if you can outcompete just by a little bit, maybe it's survivability in the environment of the female reproductive tract, maybe it's just the rate at which you get there. If you can outcompete or get there faster, maybe that's the information you will pass on. And I don't have time to go into this, but we've actually begun looking at this in human beings, again, going back from the mouse and the cells to the humans. We see all kinds of great things. This is a healthy human subject study looking at the composition and cargo of the human sperm. We found timing where perceived stress across undergraduates and medical students at the University of Pennsylvania, assessing their perceived stress and paying them to come in and give us a sample every month of their sperm. We were able to assess in a very complicated mathematical modeling that actually it was time following perceived stress, around 2 to 3 months, You started to see across all subjects these beautiful changes in the cargo in their sperm that are related to implantation and fertility. And so the latest piece of data that I can share with you, because the patent we just filed, so now I can tell you, is we began assessing this in a new cohort of individuals where Nicole began assessing the perceived stress timing again, but asking about sperm motility. And in fact, what you can see if you look across time is that there's a really nice assessment at that same time window, 2 to 3 months following the perceived stress event for these individuals. And remind, they're being recruited at constant, right? So it's not like they're all experiencing the same event or coming in at the same time, right? So this is really beautiful modeling that Nicole is working on. So we're super excited about how this might be informative again for what it means ultimately, because I am a neuroscientist, to the developing brain. So looking at this time that we consistently see in both animal studies and human studies. So I'm just going to leave you with that. I know we're a little over of time in summary, but really begin across mammals, across disease, thinking about what these EVs can tell us. We're doing all kinds of studies with our collaborators on campus related to gestational diabetes using the sheep as the model. Sheep are an incredible model for pregnancy because you can look at really specific differences at the intrauterine arteries and veins and ask questions about what's coming off the placenta. And we're also doing assessments in healthy subjects related to the correspondence of saliva EVs with blood. Because if they correspond, that would give us really a rapid assessment of individuals experiencing adversity or trauma. Anyway, so I need to thank, I mentioned all of these individuals, incredible collaborations. Please follow us on Twitter. We do all kinds of stuff important for the community and stress and trauma work. We have incredible collaborations ongoing, as I said, in several of our grants that we're doing both on campus in Colorado and with Tanya Jovanovic in Detroit. And before I walk away, before we answer questions, which I'm more than happy to do, I want to give a shout out. I am the president of EBRO, the International Brain Research Organization. This is how we fund neuroscience initiatives, schools, travel, workshops, programs, mentoring globally. If you are a neuroscientist, you are part of EBRO. There's many organizations within the Latin American region, including the Mexico Physiological Society, I believe is on here. Your fearless director has just been elected to the Latin American Regional Committee. And that is fantastic because there are now 2 individuals from Mexico who are members of that regional committee. Regional committees are the way that EBRO, from a bottom-up way, allows each region, there are 5 that encompass the world, get to determine what your priorities are locally. So I'm super excited to be doing more things to engage with Mexico neuroscience. I think this is going to be a fantastic way to have Dr. Morales be part of this community and really advocate for all of the incredible neuroscience that you're all doing, whether it's at the undergraduate level all the way up to faculty. Again, mentorship, programs around schools. If you want to travel abroad and do a study program, if you want to return home and have a way to fund your research lab. These are all things that EBRO funding can do. Please, please, the only way that we are able to do this is that all of our revenue, that 100% goes out into the global neuroscience community. It's around 4 million euro that we get. It is because of publishing in our 2 EBRO journals, Neuroscience and EBRO Neuroscience Reports. When you publish in them, it generates revenue for us to put it back out into the community. So if you are trying to decide where you want to publish and it's in sort of this domain, please consider supporting EBRO. And again, if you have initiatives or things you think might be interesting for the Latin American Regional Committee and or engagement of Mexico neuroscience, please talk to your regional committee member who is newly elected and I'm sure will be a champion for Mexico neuroscience. So I'm happy at that point to, let me go back to this, I'm happy to take any questions. I was told to remind everybody to please use the microphone because people are on YouTube.
Hi.
Hi.
Thanks for
Buenos
a
dias.
very impressive talk, regarding all the proteins, all the different proteins present in EVs, what do you think is the proportion of them that have signaling functions? Are
Ooh,
some
good
of them
question.
signaling
Yeah.
and the rest of them are small fry? Or what do you think
Yeah.
about this?
Okay, so one of the challenges in the EV space right now, despite all of the incredible research that has been done by cancer biologists and now other types of scientists, is really tools to understand. You know, when I tell you that so many tissues secrete EVs and that they can act locally and they can act at distant sites, we lack right now good tools to follow them. And so people have tried making GFP specific, so conditional transgenic animals or transgenic lines to follow them so they could ask this question about where do they go and how do they act. The problem is that nanoparticles are really tiny and GFP is really big. And so that doesn't work very well because it affects how EVs are secreted and certainly affects how they interact. So it's a fantastic question. We understand some of the key proteins, and I think I showed you some of those proteins that are important within a cell to determine how an EV gets packaged and secreted in the type of EV. How it interfaces, for instance, with the sperm is something that is part of our patent of identifying protein-protein interactions and how they might allow EVs either to provide the signal that is carried to the egg. It's a fascinating question in the intergenerational space. This is where the field is right now. Okay, so sperm can't just keep taking up these EVs in terms of cytoplasmic space. That's ridiculous. That can't happen. So is it that they hook on to the sperm through a protein-protein interface? Is it that they carry a signal? Because I showed you the EV protein interaction that happens in the caput, but then they enter the cauda of the epididymis where they're stored, and then they're ejaculated, and in the point of fertilization, they're traveling through the female reproductive tract. So the cauda produces EVs, the prostate makes a lot of EVs, the female reproductive tract makes EVs. And so it's a lot of communication. And I think where the field has figured out that there's particular times when the germ cell is more vulnerable to that signal, certainly in the caputs, that's important. But do they store the signal? Is it an interface where then they ignore other signals? And this is something that is brand new, sort of an examination in this field. We think that at the point of fertilization, the egg, there's millions of sperm that are ejaculated, right? At the point of fertilization, there's thousands. Only one is important for the point of fertilization, but thousands of sperm show up and interact at the egg. There's a lot of processes that are required to allow that one sperm to actually penetrate, right? Those EV interactions are probably really important. We just haven't got the tools yet to address it. So in some of these other studies related to like gestational diabetes, et cetera, I think that, and cancer, they're getting at more of which proteins. But you're right at the cusp of which proteins and how do they interact. It's part of how they get taken up. It's the specificity, right? They're traveling in circulation. So the protein-protein interaction is really important for which tissues will take the EVs up into the cells. But beyond the signaling there is kind of where the field is.
Thank you.
Yeah.
Hi.
Hi.
Thank you for your presentation. I want to ask you if the amount of events of sexual assault was correlated with the results.
Oh, that's a great question. We were not powered to ask that question. So if it happened, so if you were molested for years versus a single sort of a rape episode, yet it is an incredible question, especially for asking about the level perhaps, but it would have to have involved bringing the individuals back, recruiting them back in, and looking at sorts of other types of interpretation of not only the length or the timing, but also the severity of what that traumatic act. It is a fantastic question, and I think that we'll be able to address that now in our new cohort which were much more powered. So we have both men and women who have been followed for 30 years. So we'll be looking at types of trauma, not just interpersonal violence, and sort of that severity question. But you know, individual to individual, that severity question can be quite different. And so, right, because a lot of it depends, you know, I told you that women who experience adversity during adolescence are the strongest predictors of affective disturbance in the lifespan. That's true unless you have really good social support. And social support has been shown, so if you have a really tight family or social network, or you got a lot of treatment or therapy, can sort of erase some of those effects. And so we'll also know that in this 30-year cohort, I think, to assess. We'll be much more powered with hundreds. I think there's something like 500 individuals in this study.
Okay, thank you.
Yeah, it's a great question.
Hi. The context that you gave, it's fantastic, relating the stress and neurological disorders. I think it's very exciting and motivating. But I was a little bit confused with the data on the sperm, because I thought that after stress, you would get reduced ability of the sperm cells to perform. And I thought I understood that the motility, right, was increased.
Yep.
So that, how can that make sense with the idea that under stressful condition, reproduction or fertility goes down?
Yeah. So
thank you.
That's a great question, and I think that the clarification is that uses the term the allostasis of the cells. So during the stress experience, and we see this across all of our models, whether it's animals or cells, and what the epidemiological studies show us, during the stressful, chronic stressful experience, so during war, fertility goes down. During famine, of course, fertility goes down. It's when the resolution of that stress or when the new allostatic set point is met in the cells. So during the stressful experience, cells are responding to those changes in glucocorticoids, the other stress hormones in the cell. When the stressful event ends is when the cell rebounds to a new set point. And we don't see, so if we breed our animals during stress, they don't pass on these effects. When we breed them 3 months, the males, 3 months after the stress has ended is when the effect is passed on. So it's at the level of molecular allostasis in the cell that has now been achieved when the stress is over that changes the cargo of the EVs that are now interacting with the sperm. So they're carrying a new signal that enriches or enhances the fertility and the implantation is what we see.
Could that be studied at the cellular level in terms of exposing them to glucocorticoids, which I think was the experiment?
So all the studies that I rushed through and didn't tell you about, which we've been doing for the past 10 years, that's exactly what we've been doing. So we've been producing both in vivo doing stress in mice and in vitro using glucocorticoids in the epididymal epithelial cells in culture, that's stress in a dish. You can ask which things align. So that was that hugely complicated slide I didn't have time to go into.
Right, but to address what you're saying, wouldn't you expose them to glucocorticoids and then remove the glucocorticoids for a certain time?
In vivo.
In vitro.
So in vitro, so that's exactly what we did. All of those studies were glucocorticoids in vitro.
But they were more motile.
Right, so we exposed them, we exposed them to the glucocorticoids until we matched that the cells themselves and the EVs looked exactly like what we saw in vivo, and then we removed the glucocorticoids and continued to wash the cells till we reached that allostatic set point. So there's no longer, so it is exactly the same as in vivo. So we've exposed them, sorry that wasn't clear. We've exposed them until we reached an allostatic set point by removing the glucocorticoids, and now the cell is at a new metabolic set point. So Nicole has done a ton of work around the mitochondria and the nucleus. So I showed you some of her, the H3K27, the cut and run. That was all after the stress is over. The cells have been washed over multiple cell changes to show those differences. And now we collect the EVs.
Okay.
And those are the effects we saw. Sorry, that wasn't clear.
Thank you.
Yes, it's a great question. Oh, hold on, we have questions online.
One last question. regarding